Yes, Priya. So let's continue with molecular basis of inheritance. In the previous session, we had we had started with that lesson on molecular basis of inheritance, right? And we had discussed about the discovery of something called nucleic acids. This is a lesson that completely falls under the field of molecular biology, right? We are going to discuss in detail about the nucleic acid, the different types of DNA, RNA, their functioning, their regulation, all that in depth concepts we are going to learn, and we are going to learn it in detail for prokaryotic as well as eukaryotic cells. Correct? So everything has been discussed under the nucleus inside the cell. So we have seen that this entire topic of nucleic acids, DNA and all, okay, like how it was discovered. It's not an overnight thing done by just one person. Like it's a huge task and slowly things were coming up. Now also the discoveries are being made. Many more new things are being discovered. And I have told this is something which the many experts and researchers from the life sciences background, from the molecular biology background, plant sciences, animal sciences, microbiological sciences, there are people coming up together under one heading called biological sciences. Because this is something which is not restricted to only plants or only animals or only human beings. It is for every living cell. Correct? So that's why it comes under general biology topic, molecular biology. These are very concepts and very important to learn. Okay, so we have seen after the nucleic acids were discovered, people came to know that there is something called nucleic acid, the DNA, and its chemical composition. Of course, the Watson-Crick model was put forward in 1953. How the DNA structure is, that we have already learned that, right? Then we saw three experiments last time. The main common conclusion of the experiments was that there is a process called transformation which takes place, and
Transformation.
principle is the DNA. It is the DNA which undergoes transformation. That means the first one, Griffith's experiment, had proved this point. But then there was a confusion whether the transformation is because of DNA or RNA or proteins. So by the two other experiments, it was confirmed that DNA is the only transforming principle. So we are discussing, right?
Yes.
The next part of this lesson, we have to understand something really very interesting. Okay, and that is about how the packaging is done. DNA packaging. Yes. Okay. Imagine, suppose if you want to give a gift to one of your friends. Okay. And with every gift, of course, you will do some very interesting packaging.
Yes.
To your friend, it should look good, right? It should be appealing. कि yes, gift दे रहे हैं, especially when you go on birthday party में गए या कहीं पर and you're presenting it occasion. So packaging जरूरी है, and according to gifting, the packaging is decided. If it is something brittle or breakable, so you do that extra layer of packaging, dry grass या newspapers वगैरह डाल दे so that it doesn't break.
Yes.
It's a small thing but very precious, so you do that small packaging. If it's a large one, the packaging is also big. So packaging is something which we come across in our daily life always.
Yes.
अभी जैसे कॉलेज जाना शुरू करोगी ऑफलाइन, so your mom will give you some tiffin and pack it and give it to you.
Yes.
What is being given in the tiffin, the packaging is done accordingly.
Yes.
जैसे कुछ खरीद के लाती है तो पैकेजिंग होता है. Packaging is important in our daily life. Then imagine how our cells they do the packaging. किसका पैकेजिंग करना है? सेल्स में किसका पैकेजिंग करना है? DNA का. Yes, so that is the next part we are going to see. Very interesting. See, we are going to talk about the packaging of the DNA material inside the cell. Why it is important? See, it sounds very simple कि क्या है DNA को अंदर डाल दिया सेल के, but it's not that easy. Okay, see, the thing is a DNA double helix which was put forward by Watson and Crick that we know. Okay.
Yes, correct.
Mammalian cell, we are not talking about every species because सबका अलग अलग होता है, but in a mammalian cell DNA का approximate length is around 2.2 meters.
Oh yes, 2.2 meters.
Inside one cell, if I open the nucleus and if I take out the DNA and I keep it open, its length will be around 2.2 meters, more than
2.2. Yeah.
my height.
Hmm.
इतना लंबा डीएनए कॉइल करके सेल के न्यूक्लियस के अंदर डालना है.
Yes.
Getting it? So it's like
Getting it.
approximate size of a nucleus. Cell का nucleus.
Yes.
Approximate size is around 10 raised to minus 6 meter.
10 raised to minus 6 meters. Yes.
10 raised to minus 6 मतलब 0.000001 meter.
Yes.
इतने से छोटे से न्यूक्लियस में you cannot even imagine how tiny it is. Obviously not visible to the naked eye.
Yes.
उसके अंदर मुझे 2 मीटर लंबा डीएनए डालना. That's the challenge.
Yeah. Yeah.
Getting it? That's the reason in a cell this DNA is highly condensed, okay, coiled and supercoiled.
Coiled and supercoiled. What is the difference?
Condense करके छोटा कर दिया उसको
हाँ.
किया and then उसको और supercoil किया so that it is squeezed inside the nucleus.
Okay, supercoil. Okay, समझा.
Challenge.
Yes.
Okay, so how this packaging is done for the prokaryotes as well as for the eukaryotes. Let us take a look at them differently. दोनों का packaging method अलग है. Okay, so in the prokaryotic cell, now you know very well prokaryotes. इसमें आएंगे example bacteria.
हाँ.
Bacteria and cyanobacteria, they come under prokaryota, that is kingdom Monera. You know that, right? So if you consider a prokaryotic cell like say the bacteria E. coli.
E. coli.
बैक्टीरिया जानती है ना, Escherichia coli. Okay, इस बैक्टीरिया के सेल का साइज is approximately 2 to 3 micrometer long.
2 to 3 micrometer long.
Yes, 1 micrometer is 10 raised to minus 6 meter. 2 to 3 microns long. And you know very well it doesn't have a proper well-defined nucleus. Nuclear.
Yes.
That's why we use the word nucleoid.
Nucleoid. Yeah.
So it is not having a proper well-defined nucleus, but in that nuclear matter or the nucleoid which is there in the cytoplasm, there is a naked ring of DNA present.
Yeah.
Okay, and this DNA का जो length है, that is around 1100 micrometer long.
What? Okay.
Now you got the challenge for a prokaryote that the size of the cell is around 2 to 3 microns, but DNA ka length is around 1100 microns.
Oh no. Okay, got it. Got it.
Structure of the DNA, Watson Crick model which you have studied. Okay.
Yes.
It is made up of nucleotides. Each nucleotide consists of 3 main components. याद है? याद है?
Yes, I remember.
Yes. 5 carbon sugar. याद है?
Yes, I remember.
There is another phosphate group.
Phosphate molecules. Yeah.
So successive nucleotide. याद है?
Yes, I remember.
Okay, so ये पूरा unit है, उसको बोलते हैं nucleotide. So there are many such nucleotides, and on one side carbon atom number 1 के साथ connected है nitrogenous base. So there are 4 types of nitrogenous base: adenine, guanine,
cytosine, thymine.
Okay, DNA. 4 base. ठीक है? Any one of them will be present over here. Right? So that makes up the DNA molecule.
Yes.
Okay, in this prokaryotic DNA, there are 4.6 million base pairs.
What?
मैंने बोला ओके, टू स्ट्रैंड्स के बाजू बाजू के न्यूक्लियोटाइड को एक बेस पेयर बोलेंगे.
Okay.
It always combines with T. C always combines with G. याद है? Complementary base.
Complementary base pairing. Yes. Yes.
तो complementary base pairing के हिसाब से there are 4.6 million base pairs in the DNA present in the nucleoid of E. coli.
Okay, got it.
4 मिलियन बेस पेयर को 2 माइक्रोन लंबा न्यूक्लियस में फिट करना, which is around 1.1 millimeter. Okay, so क्या करते हैं DNA. Okay, DNA is a negatively charged molecule. ये point याद रखना.
Yes. Negatively charged.
Negative charge. Because the negative charge is because DNA में होता है phosphate.
Yes.
Phosphate is PO4 minus, negative radical. So that gives the DNA a negative charge.
Yes.
तो ये जो negatively charged DNA है, it becomes a circular one in prokaryotes.
Circular?
गोल घूम जाता है, circular molecule बन जाता है.
Correct. Yes.
Which the size 1100 micrometer से कम होकर it brings to 350 micrometer in diameter.
Yes. Yeah.
Okay. Then this circular DNA undergoes something called folding या looping.
Folding and looping. Yes.
जिस वजह से और छोटा हो जाता है, it comes down to 30 micrometer.
Yeah. Yeah.
अब ये जो looping हुआ, इस तरह से 40 to 50 such loops are formed.
What?
उसको भी और coil की 40-50. Getting it?
Oh.
There are something called RNA connectors. They help in forming the loops.
Oh. Okay.
Called packaging. Once the packaging is done, it has to remain stable also. अचानक से खुल नहीं जाना. Getting it? Otherwise, that's one kind of a gift that we give it to friends sometimes. कि जैसे box खोला तो pop करके emerge होगा, याद है?
Yeah. Yeah.
ये DNA ऐसे एकदम खुल जाएगा तो it will be a problem, right? तो वो नहीं होना चाहिए. तो RNA connectors help in the formation and also keep it stable. So ये coiling and super coiling, उसके वजह से the diameter comes down to 2 microns.
What? What?
And 100 microns, 2 micron पे लेकर आई, imagine.
Okay.
Now there are something called histone proteins.
Histone protein. Yes.
Type of proteins है which are positively charged, so they help in this coiling and packaging of the DNA.
Yes.
Okay, so RNA connectors क्या करते हैं? They help in forming the loops. DNA binding proteins which are histone proteins, they are positively charged, they help in the coiling.
Yes.
Okay, and there are enzymes, you have to remember the name, DNA gyrase and DNA topoisomerase 1. Topoisomerase 1, sorry.
What is this?
Enzymes के नाम है DNA gyrase and DNA topoisomerase 1. These two
Topoisomerase
1. These two enzymes they help in maintaining the supercoiled state.
अच्छा, okay.
There are 3 main steps. पहला तो है forming the loops. Coiling.
Yes.
Supercoiling.
Yes.
These steps, there are some things which are helping. Okay, loop formation RNA connectors. Okay, coiling and maintaining its stable histone proteins.
Is histone proteins or
enzymes, DNA gyrase and
DNA
topoisomerase
1. Okay, got it.
अब ये डायग्राम में देखना, ये अच्छे से समझेगा. See, it's DNA. ये circular DNA बना.
Okay.
Microns.
हाँ.
Single directly starting points. ठीक है, 1100 माइक्रोमीटर होता है, बट वो नेगेटिवली चार्ज होने की वजह से it becomes a circular molecule.
Correct. Yes.
Okay. This circular one, अब देखना उसमें क्या हुआ, foldings या loops. Can you
Yes.
see how the loops are formed? That circular one. तो
Yes.
loops की वजह से it becomes 30 micron.
हाँ, correct. Yes.
40 to 50 such loops. ये सब numbers याद रखना है. Numbers and dimensions सब बराबर याद रखना है. Okay. ये अब इसका होगा सुपर कॉइलिंग.
Super coiling. Yes.
लूप्स को और कॉइल करके ऐसा सुपर कॉइल की.
हाँ.
डायमीटर कम 2 माइक्रोमीटर दिख रहा है.
Yes. Yes.
Okay. So each loop is independently super coiled and then folded. Okay. So sometimes the coiling is partially uncoiled. तो ऐसे partially कि you can see बाकी यहाँ पे loops बना हुआ है पूरा, but at some places the DNA is open.
Oh हाँ, yeah.
तो जहाँ पे open है वहाँ पे partial DNA's digestion will take place या partial RNA's digestion will take place.
Okay, okay, got it.
That's how the packaging is carried out in prokaryotes.
Okay, got it.
अच्छी तरह से prokaryotes में कैसे सेल के अंदर.
Yes.
So that was about prokaryotes, how the DNA is packaged and kept inside the nucleoid. Okay. Now the thing we have to understand is about eukaryotes.
Oh, eukaryotes. Yeah.
कैसा है कि there is a well organized nucleus having a nuclear membrane. Correct. So nuclear membrane का मतलब है nucleus का size एकदम fix हो जाता है.
Yes.
Nucleus, the entire DNA has to be coiled and placed. And compared to prokaryotes, eukaryotic DNA is longer.
हाँ, correct. Yes.
Right. So यहाँ पे कैसा है कि there is a nucleosome model of DNA. We will come to that. But how the packaging is done in the case of eukaryotes, that was reported by somebody called R. Kornberg in 1974.
R. Kornberg. R for?
In 1974. Explain कि कैसे रखा जाता है DNA.
Yes.
Now see, in the case of eukaryotes, okay, basically, see cell के अंदर जो protein होता है, okay,
Yes.
that protein is made up of amino acids, and there are 20 amino acids in all. Each amino acid molecule has got something called side chain.
Side chain. हाँ.
तो साइड चेन में जो भी चार्ज है अमीनो एसिड का, according to that the protein gets a respective charge, whether it is a positively charged or a negatively charged.
Negatively charged. Yes.
Now we have spoken about histone proteins in the prokaryotes.
Histone proteins. Yes.
अब histone proteins के स्ट्रक्चर में जो अमीनो एसिड्स हैं, there are very high amount of 2 amino acids in the histone proteins. वो है lysine and arginine.
Yeah, I know lysine.
Yes, 20 amino acids. You know the names, right? तो histone proteins का स्ट्रक्चर अगर हम देखेंगे तो उसमें lysine and arginine amino acids are very abundant.
Okay, okay.
Both
Okay.
these amino acids are basic amino acids,
Okay.
and that's why they carry positive charges. So histone proteins, they are positively charged basic proteins, mainly because of these 2 amino acids.
Okay, got it.
Okay, histone के जो proteins है, there are 4 types of histone proteins called H2A, H2B.
H2A. H2B.
Histone proteins के type है H2A, H2B, H3 and H4.
H4.
H2A, H2B,
H2B, H3, H4.
ये 4 histone proteins है which are positively charged. In सब के 2 molecules. So you can see H2A ऊपर नीचे, H2B ऊपर नीचे, H3 ऊपर नीचे, H4 भी ऊपर नीचे. So there is a set of 8 such histone proteins, which we call as the histone octamer.
Okay.
Octa मतलब 8. Octamer.
What does octamer means?
Set of 8 such molecules of histone proteins.
Oh, 8 octave. Yeah.
4 times. Can you see that?
Yeah.
इसलिए उसको बोलते हैं octamer histones.
Oh, okay. Yes.
H2B, H3, H4, 2 molecules each. Got it?
Got it.
Okay.
Histone octamer.
Yes, ये histone octamer का meaning समझ जा. Now the thing is, see, in a eukaryotic cell, there is something called bead and string model. Okay, I'll tell you what is that. Bead and string model करके is somewhat like this.
What is that?
Okay, ये क्या है पता है? See, listen. This is the set of histone molecules. Octamer. तो ये octamer है histone का.
Okay.
Got it? तो जो तो उसको के अंदर it will appear in the form of a bead. And उसके आसपास हाँ उस in the form of a bead. Necklace.
Bead. Okay. Yes.
के आसपास you can see the thread-like structure.
Yes.
That is this DNA. Red line दिख रहा है?
Oh. Okay.
DNA molecule, it comes and coils around the histone and goes to another octamer, again coils and moves back. तो यहाँ पे देखना DNA molecule ऐसे आया,
How cool.
पूरा coil करके यहाँ से निकल.
Oh, समझा मेरे को. Okay, got it.
You understood?
Nice. Okay, got it.
2.5 turns.
2.5.
वो turns जाने दे. It could be either 2.5 या 1 and 3/4. 1 and 3/4.
Okay.
1 and 3/4 से turn करता है and then निकलता है. Okay, so at that point where it enters and the point where it leaves, there is another protein present. ये जो green में दिख रहा है.
Yes, I can see it. Green.
That is the H1 histone protein. One molecule.
H1 histone protein.
Yes. समझा उसका पैकेजिंग कैसा है?
Yes, समझा.
8 marbles of histones. Okay, one thread coming and coiling around all 8, leaving back. All of them, you can see the H1 histone, which is. So
Yeah.
totally, how many histones are there?
8.
क्या से दे? Totally, how many histones are there?
4. 1.
अरे दे,
1.
then tell.
Oh.
Say totally, how many histones are there?
Where?
इस पूरे सेट में जो डायग्राम में दिख रहा है, how many histone molecules you are able to see? Think and answer.
Okay, I want this.
How many histone molecules? अरे दिख रहा है डायग्राम में. H2A, H2B, H3, H4 कितना है? How many are there? H2A, H2B, H3 and H4. How many are there?
Oh, I can see you. Hi.
Yeah, बोल.
8, no? Because
Correct.
octamer.
8.
Yes, exactly.
H1 कितना है?
1.
तो टोटल कितने हिस्टोन्स हो गए?
9. Okay. वो तो मैंने सोचा ही नहीं.
So whenever a question is asked, first you think. You have learnt about control and coordination, right?
Yes.
CNS को पहले CNS तक पहुंचने दे, association neuron में processing होने दे, let the response come via the motor neuron and then you open your mouth.
I'm writing the text.
है ना, वो होता ही नहीं है. यहाँ से stimulus, यहाँ से response. Now, ये जो हमने discuss किया कि this DNA which is wrapped around the histone octamer, ये जो एक पूरा structure बना है, we call it as one nucleosome. ये word याद रखना.
Nucleosome.
Nucleosome.
What is a nucleosome?
ये बता रहा हूँ ना. There is
Chromosome or nucleus itself.
do not get confused, Supriya. इधर उधर
No, I'm
distract.
not. I just guessed it.
Listen. H2A, H2B, H3 and H4 के दो ऑक्टामर हो गया. एक डीएनए का थ्रेड हुआ एंड एक H1 प्रोटीन हुआ. ये सब 9 हिस्टोन्स एंड 1 डीएनए सब मिलाकर बनता है एक न्यूक्लियोसोम.
Oh, okay.
So for that,
Okay.
listen patiently and retain what is being told. Yeah,
Okay.
popcorn. Okay.
Yes.
That's it. So
Yeah.
that's a nucleosome. Now, this one single nucleosome, जिसमें DNA आ रहा है, coil करके निकल रहा है. As I told you,
Yes.
1 and 3/4 terms, not 2 and a half. Sorry. 1 and एक पूरा,
Oh,
3/4.
okay. Got it.
Got it? It's like I'll
Yes.
say, if this is the octamer, suppose. Getting it? तो ये देखना डी क्या कर रहे हैं? एक पूरा टर्न किया.
Oh, nice.
Four.
Yes. समझा.
Now you understood that? Okay.
That's
तो
like
पॉइंट पे एंटर किया जिस पॉइंट से निकला. There is next one histone protein present. And जो बाकी का डीएनए है, that will go to another nucleosome.
Oh, okay.
Reads
Okay,
on
okay.
string करके एक मॉडल दिखेगा. So between this nucleosome and between the next nucleosome, ये जो बीच का डीएनए है ना, that is known as linker DNA.
Wrinkled. W.
Linker, Linker,
Link.
Linker, linker, wrinkle का बोल.
Linker.
Yes. Okay, between
Oh, linker.
nucleosomes, the DNA is called
Okay.
linker DNA. Got it?
Got it. Yes, got it. Yes.
एक न्यूक्लियोसोम है. इसमें जो डीएनए प्रेजेंट है that is around 200 base pair long.
Oh.
And it enters the nucleosome 1 and 3/4 and it leaves. So it is 200 base pair long. Got it?
Got it. Got it. Got it.
And around 146 base pair remains present in each nucleosome. जो उसका एक्स्ट्रा थ्रेड पोर्शन छोड़ के जो एक्चुअल न्यूक्लियोसोम में है, that is 146 base pair long.
Okay. समझा.
That's why it gives rise to beads on the string model, which is seen under electron microscope. ये देखो.
Beads on the string.
Yes.
Oh, how cool. Show us. Okay.
ये दिख रहा है न्यूक्लियोसोम्स कैसे एकदम ऑरेंज.
Yes.
तो इस तरह से बीड्स एंड स्ट्रिंग बन के जो पूरा स्ट्रक्चर नजर आता है, which is seen under electron microscope, that gives rise to one chromatin.
What?
Yes, that gives rise to one chromatin.
Oh. Okay. Got it.
अब देखना, there are 6 such nucleosomes. Okay, which forms one solenoid. 6 ऐसे nucleosomes, it will form a solenoid which is coiled like a telephone wire.
Oh.
वायर देखा जो लैंडलाइन फोन होता है, you pick it up and there is a wire spring जैसा.
हाँ.
Yes,
हाँ.
correct.
Yeah.
Solenoid. So whenever you are using a landline phone, उसका वायर देखने का, रिवाइज करने का, it's a solenoid. Getting it?
Okay. समझा. Yeah.
Solenoid का diameter is around 30 nanometer diameter, 300 angstrom. 30 nanometer. All these numbers which are coming up, इसका dimension, number, diameter, everything, you have to learn as this. All these are.
I will do that. Yeah.
फोन का वायर है सोलेनॉइड, उसका होता है सुपर कॉइल, जैसे पहले हमने देखा.
Oh.
It's a super
Okay.
coil.
Okay.
So that super coiling, when it happens, it becomes a chromatin.
Oh. Now I got it. I got it.
In a eukaryote, there is a nucleosome okay, which is one bead, many nucleosomes forming bead and string. 6 of them together is one solenoid. That solenoid undergoes coiling, supercoiling, giving rise to chromatin. तो सेल के अंदर न्यूक्लियस के अंदर जो न्यूक्लियोप्लाज्म है, in that the highly coiled structure that you get is the chromatin. So just before cell division during the interphase, this is the thing which is condensing to give rise to chromosomes.
Yes, got it.
And
Yes.
packaging. There are other proteins also involved at a higher level. Chromatin के packaging. They are
Yes.
proteins called non-histone chromosomal proteins. They also
Non-histone chromosomal protein.
got it.
Got it. Oh.
As I said, packaging aim क्या है? See this. This is nucleosome. This is linker DNA as also.
Oh, wow. Okay, got it. Got it.
This
Got it. Yes.
main aim क्या है? It's not just to coil, supercoil and put it in the nucleus. उसका दूसरा main function है to keep it stable.
Yes, correct.
Functioning also. इन्हें काम भी तो करना है ना, सिर्फ ऐसे घुसा के नहीं रखा. That is a function. Okay. So जो linker DNA है, its length is around 8 to 11. 114 nucleotide pairs. That's the length of a linker DNA. All these numbers you have to learn. Okay. So the chain of nucleosomes, they form 10 to 11 nanometer thick fiber. That is also formed because of the nucleosomes. ऐसे करके ये सब उसका solenoid, 30 nanometer thick fiber, all this you have to learn, which will help you to understand how the DNA is ultimately packaged inside the cells. Is it clear? So you have understood properly how the packaging is carried out at the eukaryotic cell also, based on the string model, nucleosome, solenoid, chromatin, chromosome. Okay.
Chromosome.
The last point that we said that there's additional set of proteins which are present called NHC.
NHC.
Histone
Non-histone
proteins.
chromosomal protein.
Yes. Okay. At a higher level, they also help in packaging. So that NHC is of two types. You have heterochromatin and euchromatin. Okay. NHC, additional set of proteins. They are of two types. You have heterochromatin and euchromatin.
Euchromatin.
Okay.
Okay.
See, it's like, see, in a eukaryotic cell, during the interphase, some of the chromatin material, they remain in a condensed state or inactive state.
Inactive state. Oh,
Inactive state. तो ऐसे जो regions होते हैं उनको बोलते हैं heterochromatin. कि they are present in the cell but they are inactive. They are condensed. And this term was provided by a person called Higgs.
Higgs. His name was Higgs.
Higgs.
Yeah.
Okay. So you have seen the structure of a chromosome in the earlier lesson. You know something called centromere, telomere.
Yes. Yes.
Right? जो heterochromatin होते हैं, they are generally located in these centromeres and telomeres. Okay, genetically very active, and that's why whenever a cell is stained and observed under the microscope, they appear very bright. They stain very strongly and appear very dark.
Oh, okay.
Heterochromatin, it is 2 to 3 times more rich in the DNA as compared to the other one, that is euchromatin. Euchromatin is the opposite one. It is genetically very active and fast.
Oh, okay. Okay. Oh, I got it. It is easy. Okay, I got it.
They do not take up a lot of stain. They stain very light. So it's in a non-condensed state in the cell.
Yeah.
So that's why euchromatin is the one which remains functional and which undergoes transcription followed by translation.
Oh, okay.
inactive. Getting it? In this context, there is one more thing I would like to tell. Like in the case of human beings, okay, you know, we have 46 chromosomes.
Oh yes, I know we have 46 and 23 pairs.
Yes. So 23 pairs जो होते हैं, one pair of chromosome we call as the sex chromosomes.
Yes.
XX in a female, XY in a male.
XY in a male, yes.
Means females have got one more extra dose of X chromosome.
Yes.
In a female, that one extra X chromosome का जो major region है, that is in a condensed state. That's why in human beings, in a cell, if you observe under the microscope, you will see that अगर ये cell human beings का लेता हूँ, तो you will find darkly stained portion inside the cell if it is taken from a female.
समझा नहीं? Can you repeat?
See, एक experiment किया जाता है, okay?
Yes.
It's like what one has to do is using a soft cotton bud, जो inner lining of cheeks है, buccal lining बोलते हैं, okay, inner lining of the cheeks, the buccal lining, वहाँ से एक scraping लेना होगा, just okay, not even a scraping, just a smear you can see, जिसमें यहाँ के cells collect हो जाए, and then that is spread on the microscopic slide. Cleaning करके microscope में देखा जाते हैं. So if the lining is taken from a woman or a girl, so you will be able to see a reduced, condensed chromosome like this. But if it is taken,
और अगर मेल है तो
नहीं दिखेगा.
दिखेगा ही नहीं. Okay.
तो ये जो होता है, which is observed only in a female, उसको बोलते हैं bar bodies.
Bar bodies.
One who discovered it, उसका नाम था bar.
उसका नाम बार है. BRR, 2R.
Yes.
Okay.
So that's why they are called bar bodies. So they are nothing but the heterochromatinized X chromosome, which is extra in a female. Males में XY होता है ना, extra X तो है ही नहीं. That's why no bar body is male. Have you understood?
Understood.
Chromatin and euchromatin. Getting it? So bar bodies can be observed when the specimen is collected from two locations. One is the inner lining of the cheeks, the buccal lining, and second is the vaginal smear.
Oh, अच्छा. Okay.
Just the bar bodies can be observed.
Okay, got it.
Understood the difference between heterochromatin and euchromatin?
Yes, understood.
DNA packaging is clear properly.
Yes, clear.
Yeah, good. Now let's go to the next part. Now we have to see the functions of the DNA. Okay. We know the structure of the DNA, we know the location of the DNA, we know how the packaging is done.
Yeah.
के अंदर न्यूक्लियस के अंदर डीएनए पड़ा हुआ है, सेफ है, स्टेबल है. Okay. The thing is how the DNA actually functions.
How? Tell me.
Basically, what is the function of a DNA? DNA is the one that contains genes which control the heredity.
Heredity, yes.
Which gives rise to certain characteristics. Correct?
Yes, offspring.
Yes. So when you talk about the functions of DNA, they are of two types. One is heterocatalytic function. Heterocatalytic function of DNA का मतलब है when a DNA molecule gives rise to any other molecule except DNA.
Oh, except DNA.
DNA में से RNA बनता है, DNA से protein बनता है. So it is not DNA, some other molecule is formed. So synthesis of RNA from the DNA is called transcription. Synthesis of protein is called translation.
Yes.
So ऐसा जो function है where a DNA molecule gives rise to any other molecule except DNA, we call it heterocatalytic function.
Heterocatalytic function.
The other function is called autocatalytic function.
Hetero, auto. Got it.
It means a DNA molecule is able to produce another DNA molecule itself.
Huh? Okay.
Okay,
Okay.
polyphyletic, and that is called as the DNA replication.
Replication. Yes.
Okay,
Correct.
making its own copy. So DNA
Yes.
duplicating itself is called replication.
Okay. Got it.
DNA molecule giving rise to two daughter DNA molecules which are exact copies of the parental DNA.
Yes. Correct. Twins. Correct.
डीएनए को अपना खुद का कॉपी बनाने का जरूरत कब पड़ सकता है? Why will a DNA have its own copy?
Why?
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 Why will a DNA want to make its own copy? You have learned about cell division.
Yes.
Are you able to get a hint? सेल डिवीजन में सबसे पहले जो डिवाइड होता है वो है न्यूक्लियस. Karyokinesis.
Yes.
So karyokinesis takes place. The nuclear matter also divides equally. Like mitosis that you have learned. The nucleus divides into two, but the ploidy number of the chromosome does not change.
Does not change.
46 remains 46. That means the DNA content will first become double and then separate.
Oh, yes. Yes, you're right. You're right. You're right.
So,
Yes.
if you recollect something called cell cycle that you have learned.
Cell cycle. Yes.
One
First
G,
you talk. Yeah.
one S, G2 and M, 4 phases.
Yes, correct. Yeah.
M is the actual dividing phase. But uske pehle G1, S and G2, all three are together called the interphase.
Interface. And वो होएगा
In that interphase, the cell is preparing itself for the division. Interphase में जो S phase आएगा, that is the synthesis phase. That means new synthesis of molecules are being happening.
Correct. Yes.
Phase where DNA replication takes place. S phase of the interphase in the cell cycle. Understood?
Yes.
Very, very important question. Replication of DNA takes place in the S phase of the interphase.
The interface in the cell cycle. Okay, got it.
समझा DNA replication का actual meaning and when does it take place?
हाँ.
अभी हम लोग को देखना है कि how does the DNA actually replicate? It's a very interesting ध्यान से सुनो.
Okay. Okay.
See, DNA replication जो है, it is said to be a semi-conservative method of replication.
Semi-conservative?
We will come to this point once again after you learn the entire process. Just remember this word. We call it semi-conservative. Okay? So this model, okay, how the semi-conservative replication takes place, okay, was proposed by Watson and
Crick, of course. Yes.
Okay. See, basically aim क्या है? If this is the DNA. Okay, yes, a DNA double stranded. You know the structure. So one strand is from 5 prime and
to 3 prime.
3 prime. Correct?
Yeah.
One strand is from 3 prime
to 5 prime.
Opposite in direction but parallel to each other. तो हम लोग उसको बोलते हैं parallel.
Anti-parallel. Yes.
And it is complementary.
Complementary. Yes, correct.
There should be T on the other side and vice versa.
T and G. GC. Yes.
Point है Watson-Crick model का structure में कि DNA both the strands are complementary and DNA both the strands are anti-parallel. These two are the main points which are used to understand how the DNA replication takes place. तो बेसिकली होता क्या है पता है कि in this DNA अगर इसको अपना खुद का एक कॉपी बनाना same to same तो one strand of DNA will become separate from the other. ये पहला वाला 5 prime to 3 prime दूसरा वाला 3 prime to 5 prime दोनों स्ट्रैंड के ऊपर एक एक नया स्ट्रैंड बनेगा। गेटिंग इट? तो मतलब पेरेंटल इज हैविंग टू स्ट्रैंड्स, पेरेंटल स्ट्रैंड्स, उन दोनों के ऊपर नया स्ट्रैंड बन रहा है, वो है डॉटर स्ट्रैंड। कॉम्प्लीमेंट्री ही रहेगा, एंटी पैरेलल ही रहेगा। तो ऐसा जो पूरा नया मॉलिक्यूल बन रहा है उसमें वन स्ट्रैंड इज पेरेंटल, वन स्ट्रैंड इज डॉटर। 50% of the parental strand remains as it is in the daughter molecule.
Yes.
इसलिए semi conservative. As it is. पूरा नया daughter molecule नहीं बनता। That's the main aim. तो अभी देखते हैं डिटेल में how this actually happens. But you understood how it happens basically?
Yes. Yes.
Okay? अभी क्या है, डीएनए का मॉलिक्यूल है, it's a long molecule. So entire length of the DNA need not replicate at the same time. Okay? तो उसका कोई छोटा हिस्सा बीच का पार्ट है वो ओपन अप होगा, रेप्लिकेट होगा, नया बनेगा, आगे पीछे का बाकी सेम रहेगा। So that point किधर से किधर तक रेप्लिकेट करना है। It's ultimately a single DNA can have many genes. So maybe one gene in between will replicate, others will remain as it is.
Got it.
First thing that is decided, which point ultimately the replication must begin. So उसके बाद दूसरा important requirement आता है कि replication करने के लिए the two parental strands which are coiled and which are complementary base pairing से attached है, उनको separate open करो। ओपन करके उसके ऊपर नया स्ट्रैंड बनाओ।
Okay.
स्ट्रैंड बनाने के लिए like suppose the parental strand is having T as the nitrogenous base.
Okay.
स्ट्रैंड बनाने के लिए A को लाना होगा।
Yes. Got it.
सिर्फ A नहीं आएगा, पूरा न्यूक्लियोटाइड आएगा। A is the 5 carbon sugar and the phosphate पूरा।
Yes.
तो ऐसे करके individual nucleotides which are the raw materials they are present in the cytoplasm या nucleoplasm.
Yes.
न्यूक्लियोप्लाज्म में बहुत सारे ऐसे न्यूक्लियोटाइड्स पड़े हुए हैं but which are inactive. So
Correct.
which one is present on the parental strand, the respective nucleotide is picked and brought and attached over here.
Oh, okay. Okay.
तो सबसे पहला जो स्टेप है, इसका बहुत सारा स्टेप्स है रेप्लिकेशन का। सी द फर्स्ट वन, द फर्स्ट स्टेप इज एक्टिवेशन ऑफ द न्यूक्लियोटाइड्स इन द न्यूक्लियोप्लाज्म। Okay, all these nucleotides of DNA, 4 types है ATCG. Okay, these 4 types are present in the nucleoplasm, but in a bound form. It is not in a free form. So, in the nucleoplasm, these 4 nucleotides are present. Okay, and as I said, whenever a DNA has to replicate and a new strand of DNA has to be formed, these nucleotides are picked up and they are placed accordingly. Okay. Nucleotides which are present in the nucleoplasm, they are not just present as molecules of adenine, guanine, thymine, cytosine.
Cytosine, yes.
Molecular form. So you can see DAMP. Now D of adenosine monophosphate.
Oh, yes.
Guanine monophosphate. D of cytosine monophosphate. And D of thymidine monophosphate. ये nucleotides which are in an inactivated form. So inactivated है तो they cannot react as such, but they have to be activated because एक साइड में DNA replicate हो रहा है तो यहाँ जरूरत पड़ रही है nucleotides को. So they need activated, for which ATP molecule is required. Energy currency. And
Yes, yes.
at every step, remember what are the molecules needed, what are the enzymes needed. ATP molecule चाहिए and enzyme होता है phosphorylase.
Okay.
To each of these mononucleotides, the ATP molecule reacts. Enzyme is phosphorylase, so you are getting active deoxyribonucleotide triphosphates. Okay, जैसे DAMP था, अब हुआ DATP. This ATP and this ATP.
समझा मेरे को. Okay, got it.
ये T थायमिन वाला ATP है.
हाँ.
एडेनाइन वाला ATP है. ये गुआनाइन वाला GTP, साइटोसाइन वाला CTP, थायमिन वाला TTP. So
okay, got it.
These are the activated form of the nucleotides.
Activated.
Yes, that process is called phosphorylation.
Phosphorylation, yes.
Phosphorylation is the first step of DNA replication taking place in the nucleoplasm in the presence of the enzyme phosphorylase and ATP molecule.
Okay.
So nucleotides.
Okay, easy. Got it.
All the nucleotides are ready. Understood that?
Understood.
Step of DNA replication. Now going on to the second step, we have point of origin, also called the initiation point. That means it is decided where the DNA replication must begin. At which particular nucleotide? There are DNA क्या है base pairs है ना पूरा. तो कौन से दो बीच में begin करना है replication? That specific labeled as O for the origin. And DNA replication must end. That is the termination point called T.
Yes.
O and T ये डिसाइड हो जाएगा कि इन दोनों के बीच में पूरे डीएनए को रेप्लिकेट करना है.
Okay, got it.
That becomes the unit of replication, which is called as replicon.
Replicon. Okay, got it. Easy.
तो प्रोकैरियोटिक डीएनए और यूकैरियोटिक डीएनए में जरा सा डिफरेंस है when it comes to replication. Prokaryotic DNA got only one replicon.
Oh.
Means the entire DNA replicates together. Eukaryotic DNA is much complex and very big. तो उसमें बहुत सारे replications होते हैं in a eukaryote. Understood the difference?
Oh. Okay, understood.
So ये process ऐसे देखा जाए ज़्यादा difference नहीं है replication में. But the replicon, which is the unit of replication, is only one in the case of prokaryote and many in the case of eukaryotes.
Eukaryote, okay.
Origin and this point is decided. तो जब decide हो जाएगा कि okay O is the origin, that point, so there is one enzyme that comes into picture, वो है enzyme endonuclease. Endonuclease.
Okay.
Where is that point on the DNA where it has to begin the replication? At that point, this enzyme will nick. Nick का मतलब छोटा सा break.
Break.
Yes. दोनों strands separate करेगा क्योंकि this is the point where it has to start.
Oh, how cool. Okay, got it.
Phosphate का जो backbone है. हाँ. न्यूक्लियोटाइड में अगर देखेंगे तो बैकबोन इज शुगर फॉस्फेट ऐसा है. And yes. Nucleotides, the bond which is present is called phosphodiester bond.
Phosphodiester bond. Yes.
That phosphodiester bond between successive nucleotide, that point is the one where the nick is created by this enzyme.
Oh, अच्छा. Okay.
Yes.
Okay. Wow. Okay, got it.
Second point है कि decide हो गया कहाँ से start करना है. जब decide हो गया कि start करना है, तो at that point nuclease has actually broken the bond. दोनों strand
broken the bond
separate हो गया. अब separate हो गया तो
समझा मेरे को. Okay. Yes.
Getting it? Like if I have a zip है तो कैसे खोलते हैं? ऊपर का एक पॉइंट पहले ओपन करते हैं, then you just keep on opening it.
Yeah.
उसी
Yeah.
तरह से यहाँ पर अभी होगा नेक्स्ट स्टेप अनवाइंडिंग ऑफ द डीएनए मॉलिक्यूल। तो एक स्ट्रैंड पे ओपन हो गया, अब उसको खोलते जाओ बोथ द स्ट्रैंड्स।
Correct. Okay.
For that the enzyme is DNA helicase. Remember the every enzyme at every step they are very specific as you can see.
हाँ.
यानी कि दोनों स्ट्रैंड्स के बीच में नाइट्रोजनस बेसिस का जो बॉन्ड है दैट्स कॉल्ड एज अ हाइड्रोजन।
Yes.
बॉन्ड को
Correct.
so the two strands are getting separated
हाँ.
and it's a DNA double helix. तो जब सेपरेट होगा तो एंजाइम इज हेलिकेस।
Helicase. Okay. Got it.
DNA from O to T will keep on opening. दोनों सेपरेट हो।
Okay.
That's why this unwinding is said to be bidirectional. Okay.
Said to be?
Bidirectional मतलब दोनों साइड।
अच्छा. Okay. 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 Got it.
What happens is
Oh.
this is the point O and यहाँ से ओपन हो रहा है, सेपरेट हो रहा है, तो एक ऐसा Y शेप replication fork बनेगा।
हाँ.
Getting it? There is a Y-shaped structure form which is called as a
Okay. Okay. Okay.
but yes, here we are talking about the two strands of DNA getting separated. So
Yes. Correct.
the strands separate हो गए, they are not stable. Why they are not stable? Because nitrogen bases will always tend to pair, no?
Oh yes.
ये तो वो दोबारा से खुद में कॉइल होने को ट्राई करेंगे to become stable.
Yes.
So that's why they called SSBP.
SSBB.
Single stranded binding proteins. ये single stranded DNA binding proteins. तो जैसे ओपन होता जाएगा वो एक एक स्ट्रैंड के ऊपर SSBP बैठ जाएगा कि अब अनवाइंड अभी वाइंड जॉइन नहीं करना, रुक जाओ। It will prevent the rejoining because जब वो बनेगा तभी उसके ऊपर नया स्ट्रैंड बनेगा ना।
Yes. Yes.
Are you getting
Oh,
it?
समझा.
Prevent
Okay.
rejoining.
Rejoining. Okay.
ऊपर अटैच रहेगा ताकि उसके ऊपर नया स्ट्रैंड बना सके। So you have to remember SSBP. The role is
Okay. Got it.
prevent
SSBB.
rejoining or recoiling of the separated strands. So when you talk about the unwinding of DNA molecule, there are two main enzymes coming into picture. One is DNA case, which separates out, and SSBP binds the separated strands to prevent the rejoining.
Okay. Got it.
You understood this?
Yes. Understood.
The same. Okay. तो जब ये separation हुआ, now you can see this is one parental strand, this is another parental strand.
Yes, I can see. हाँ.
जो दोनों parental strands हैं, we call them as the templates.
What is template? Oh.
अब है वो reference strand जिसके basis पे नया strand बनेगा।
Okay, got it, got it.
So there are two types of template produced. One is called lagging template, and the other one is called leading template.
Okay, got it.
The reason I come to that. But अभी के लिए समझ कि there are two strands which are present. Both are the one of them is a leading template, other one is a lagging template.
Okay, got it.
Okay, अभी जो leading template है, इसका leading template का direction देखना. It is 5' to 3'. Can you see that?
हाँ, yes.
इस जहाँ से ओपन हुआ है वहां से देख 3' to 5'.
Yes.
जो लैगिंग टेम्पलेट है उसका डायरेक्शन है 5' to 3'.
हाँ.
Getting it?
Oh yes.
इसके
Yes, yes, yes.
स्ट्रैंड बनेगा. One thing you have to remember, the new strand which is produced is always produced in 5' to 3' direction. 3' of
5' to 3'.
Got it?
Yes, got it. Understood.
I hope you are concentrating.
Yes, I am.
The new strand which is produced is always from 5' to 3' direction.
Okay. Okay.
पैरेंटल स्ट्रैंड 3 प्राइम से स्टार्ट हुआ है गोइंग अप टू 5 प्राइम.
हाँ.
इसके ऊपर जो बनेगा वो
Yeah.
प्राइम इधर आएगा, नया स्ट्रैंड बनेगा.
हाँ. Correct.
ये कंटिन्यूटी में बन जाता है बिकॉज वही डायरेक्शन में बनाना है. एंड 5 प्राइम
Oh
टू
yes. Got it.
दैट्स व्हाई दिस हैपेंस फास्टर.
Okay. Got it.
टेम्पलेट है where the parental strand is from 5 prime to 3 prime. उसके ऊपर जो नया बनेगा वो 3 प्राइम से 5 प्राइम नहीं बन सकते. That's why it is produced in reverse direction. 5 prime to 3 prime, 5 prime to 3 prime ऐसा करो. So compared to one, the new strand on the lagging template is produced later. That's why उसको lagging. Lagging मतलब पीछे रह गया. Leading मतलब आगे निकल गया.
Oh. Okay.
That's why it is a continuous one. This is a discontinuous one. You understood this? Lagging and
समझा.
okay. See the next step.
Okay.
Replicating fork. So there is a Y-shaped replication fork created.
हाँ. एक मिनट. आवाज नहीं आ रहा.
Hello, am I audible?
Yes, you're audible.
So ये unwinding और separation की वजह से there is a Y-shaped replication fork which is produced. Okay, and ये जो unwinding की वजह से there is a strain created. मतलब kind of instability हो रहा है. The strain which is created. खोल के रखा है, जबरदस्ती का पकड़ के रखा है ना.
हाँ.
तो रिलीव करने के लिए there is another enzyme called superhelix relaxing enzyme.
Okay.
Remember this enzyme also.
Okay, I will.
Understood this?
Yes.
अच्छी तरह से superhelix relaxing enzyme which will relieve pain which is imposed due to the formation of replication fork.
Okay.
It's like DNA में सबसे पहले डिसाइड हुआ where it has to start the replication. वहाँ पे एक nick हो गया. Then the actual binding took place. Unwinding के बाद वो separated strands को stable रखने के लिए SSBP was there.
Yes.
इस सेटअप में strain create हो गया जिसको relax करने के लिए superhelix relaxing enzyme.
Oh, okay.
The two parental strands are ready. चलो इसके तो ना जो synthesize होगा, as I said, it can only from the 5 prime end. 5 prime to 3 prime direction में ही बन सकता है. तो उसके लिए the beginning takes place with the help of a molecule which is a small RNA molecule जिसका नाम है RNA primer.
Yeah, RNA.
Primer.
Okay.
ये अच्छा इनिशिएट करेगा. So here as you can see, जैसे ये लीडिंग टेम्पलेट है, उसमें स्टैंड में 3 प्राइम है. तो लीडिंग टेम्पलेट का 3 प्राइम डायरेक्शन में आरएनए प्राइमर आके अटैच हो जाए.
Okay.
RNA primer. It will always attach to the 3 prime template. That's why 5 prime to 3 prime में ही बनता है. तो RNA initiate. Okay, and then जैसे वो strand के ऊपर आगे बढ़ेगा. According to the sequence which is present on the template, the respective nucleotide from the nucleoplasm is picked up and attached. पैरेंटल टेम्पलेट में अगर लिखा, अगर C वाला न्यूक्लियोटाइड है साइटोसाइन, nucleoplasm guanosine guanine will be picked up, that will be attached, then it will go to the next nucleotide. T है तो A को उठा के लाएगा, अगर G है तो C को उठा के लाएगा. Like that it will continue, but it will always be beginning by RNA primer.
Okay, fine. चलेगा.
जैसे न्यूक्लियोटाइड लेके आ रहे हैं उनके बीच में so there is a nucleotide strand also created.
Which strand?
There is a nucleotide new strand which is formed because one by one the
Okay.
and attached. And ये process करने के लिए the enzyme is DNA polymerase.
I know this. I know this.
Polymerase. Okay, and complementary strand
Yeah.
is formed in 5 prime to 3 prime direction.
Yes, correct. You're right. Yes.
समझ रहे हैं कैसे
Yes,
बनती
clear.
है?
I got it.
Going back to the lagging and the leading template. टेम्पलेट के ऊपर जो नया स्ट्रैंड बनेगा दैट विल बी द लीडिंग स्ट्रैंड. लैगिंग टेम्पलेट में जो नया स्ट्रैंड बनेगा दैट विल बी द लैगिंग स्ट्रैंड.
Okay, got it.
It always starts at the C3 end of template strand.
Yes.
Template का C3 तो नए स्ट्रैंड का C5.
Yes.
तो लीडिंग स्ट्रैंड के ऊपर जो लीडिंग टेम्पलेट के ऊपर जो नया स्ट्रैंड बन रहा है वो एक साथ कंटिन्यूटी में बन जाता है. 5 प्राइम में स्टार्ट होके पूरा नया स्ट्रैंड एक साथ बन जाएगा. दैट्स व्हाई इट इज अ कंटीन्यूअस स्ट्रैंड.
Oh, अच्छा. Okay, got it.
Leading template, as you can see, it is formed in fragments. उल्टा डायरेक्शन में आना है ना.
Yes.
थोड़ा
अच्छा, okay, मेरे को समझ गया.
फ्रैगमेंट्स में बनेगा. उन फ्रैगमेंट्स को बोलते हैं ओकाजाकी फ्रैगमेंट्स.
Then what, sir?
They are called as ओकाजाकी फ्रैगमेंट्स. जो टेम्पलेट के ऊपर पीसेस में बनेगा इन अ 5 प्राइम टू 3 प्राइम डायरेक्शन.
अच्छा, okay.
Fragments. Now you understood?
Okay, easy, got it.
Okazaki fragments will match. पूरा आधा बन जाएगा तो जोड़ देंगे.
Okay.
There was a scientist who discovered this, whose name was Okazaki.
Oh, of course.
Fragments बनेंगे Okazaki fragments. Then those fragments are joined, and for that the enzyme is DNA ligase.
Ligase. Oh, okay. Yes.
DNA ligases are joining enzymes. Yeah. Okay, they join fragments and ultimately जो lagging strand बना है वो भी पूरी तरह से complete हो जाएगा.
Okay, clear, very clear. Got it.
RNA primer was the one which initiated the formation of the new strand. DNA polymerase जैसे ही आके continue करेगा, RNA primers are removed by it.
Oh, okay, got it.
Getting it?
Getting it.
DNA polymerase are the ones that remove the RNA primers and replace them with something called DNA polymerase 1 in prokaryotes and DNA polymerase alpha in eukaryotes.
Alpha?
Yes. दोनों में अलग-अलग हैं enzymes. Okay, वो नाम याद है. तो ऐसे finally you are getting the lagging strand and the leading strand both.
Okay.
Joining हो गया DNA ligase से. So you are the two different strands. Parental DNA, daughter DNA strands. तो जब पूरी तरह से strands बन जाएंगे, अब उसका next role आता है. The strand should coil like a double helix, जैसे normal DNA में होता है.
Double helix, yes. Right.
For that, the enzyme is DNA gyrase.
Gyrase. Okay.
Also called as topoisomerase.
Also called as?
Topoisomerase.
Okay. Chalega. Got it.
Topoisomerase will ensure that the double helix is formed again and the two daughter DNA molecules are ready.
Okay. Simple. Got it.
You can see at the end, the 50% of the mother DNA या parental DNA remains as it is in the daughter DNA. इसलिए इसको बोलते हैं conservative method of replication.
Okay, got it.
It remains a parental strand. One other strand becomes a newly synthesized.
Okay.
Is the process clear? सारे enzymes at which point are needed. And step wise कितना minutely precaution लेकर किया गया.
Yes.
एक और चीज है कि when this DNA polymerase जा रहा है, नया न्यूक्लियोटाइड्स लाके ऐड कर रहा है.
Yes.
Whatever sequence is there on the parental template, according to that, there is a daughter template added, daughter nucleotide.
हाँ. Yes.
So जैसे ही daughter nucleotide डाल दिया, अगर maximum time it is correct कि T है तो A डाला, C है तो G डाल दिया.
हाँ.
Still, mistakes can happen.
हाँ.
तो उसके लिए डीएनए पॉलीमरेज के पीछे पीछे देयर इज समथिंग कॉल्ड डीएनए मैकेनिज्म. गलत न्यूक्लियोटाइड को लगा सी के साथ सपोज टी को डाल दिया, वैसे वो बॉन्ड होगा ही नहीं, इट विल रिमेन ओपन, बट स्टिल द डीएनए रिपेयर मैकेनिज्म विल एंश्योर दैट द इनकरेक्ट बेस इज रिमूव्ड एंड द करेक्ट वन.
अच्छा. ओके. समझ गया.
ओके, तो ये सब प्रिपरेशन के साथ किया जाता है, एट द एंड ऑफ व्हिच फाइनली फ्रॉम वन डीएनए मॉलिक्यूल टू डॉटर डीएनए.
Okay. Got it.
Exactly.
हाँ. Got it.
Stepwise process properly.
Yes. Properly.
DNA replication is semi-conservative. ये experimentally
Semi-conservative.
by two people, that is Matthew Michelson and Franklin Starr in 1958.
ये लोग कौन है? Okay. Fine. First time.
There are people, yes, Michelson and Starr's experiment करके होते हैं. Okay.
Okay.
It was called equilibrium density gradient centrifugation technique.
Oh. Okay.
हमने डीएनए का ट्रांसफॉर्मेशन प्रिंसिपल के ऊपर एक्सपेरिमेंट किया था, वेयर वी हैड हर्शी चेस एक्सपेरिमेंट, इफ यू रिमेंबर, वेयर
Oh, Hershey. Yes. Yes.
रेडियो एक्टिविटी को यूज किया था यहाँ पे.
हाँ. Yes. Of course. I remember.
सॉरी, डीएनए का फास्फोरस एंड प्रोटीन का सल्फर रेडियो एक्टिव रखे थे, सो दैट वी कुड ट्रेस.
हाँ. Yes.
दैट सिमिलर प्रिंसिपल इज यूज्ड ओवर हियर. Michelson and Stahl experiment in 1958, where nitrogen का जो आइसोटोप है, that is N14.
हाँ.
और नाइट्रोजन का रेडियोएक्टिव आइसोटोप है N15.
हाँ.
Getting it? So रेडियोएक्टिव आइसोटोप कौन से सकते हैं?
Oh, हाँ. हाँ.
What they did, बैक्टीरिया E. coli को कल्चर किए in a medium that was having normal nitrogen.
Okay.
Okay, तो
Okay.
उससे क्या होता है कि there is something called as a band which is recorded. Okay. Which equilibrium gradient band करके बोलते हैं, and 6 molar sodium chloride is used for the banding. उसके डिटेल में अभी नहीं जाते, leave it, just understand कि density gradient का band तैयार किया after culturing
ठीक है. Okay.
like nitrogen. Then the same E. coli cells were transferred to another medium जिसमें नाइट्रोजन का heavy isotope है N15.
Oh, okay.
इन वाले मीडियम में इन ई कोलाई के सेल्स को रेप्लिकेट करने के लिए छोड़ दिया.
Oh.
In N14. Now they are in N15.
अच्छा.
N15 में रेप्लिकेट करने के बाद again we got the bands. तो बैंड का पोजीशन रिकॉर्ड किया गया and there was a difference observed. See this.
Oh.
तो ये क्या है? ई कोलाई के सेल के अंदर DNA present, both the strands of DNA are having N15 isotope because radioactivity.
Yes. Of
Got
course.
it? But
got it.
this bacteria, when it was transferred to N14 and N15 condition. See, पहले
हाँ.
बाद में N15 में लेकर.
हाँ.
तो जब replication हुआ, so it was seen that when one DNA having N15 gave rise to two daughter molecules of DNA, it was seen that daughter molecules were having one strand with N15 isotope, one strand with N14 isotope.
हाँ, correct. Yes.
इसका मतलब क्या हुआ कि ये डीएनए मॉलिक्यूल्स के स्ट्रैंड सेपरेट हुए एंड एक के ऊपर नया N14 बना, दूसरे के ऊपर नया N14 बना.
हाँ, correct. Yes.
Getting it? That was generation. तो ये जो बैंड्स होता है ना, वो बैंड्स को देख के पता चलता है कि N15 है तो बैंड N14 15 हाइब्रिड है तो इधर आया. See see the N14 light one and N15 light one.
Oh, yes.
Second generation. अभी इसमें कैसा है? Both the DNA molecules are hybrids. N14 and 15.
Yes.
अब जब इसका replication हुआ, the first one if you see, so one strand is having only N14, तो उसके ऊपर जो नया strand बना वो भी N14 है, तो पूरी तरह से N14 आया.
Yes.
And जो रेड वाला है N15, उसके ऊपर N14 बना, तो हाइब्रिड है.
हाँ.
Same way for the other one. So here you two molecules are fully N14, two of them are hybrids. तो बैंड देखो कितना N14 की तरह.
Okay. समझ गया मेरे को. Okay.
That's why equilibrium density gradient बैंड करते हैं.
Okay, got it.
वो जनरेशन में देखने के बाद it is clear that one strand of DNA remains the same as that of the parental one, and one other strand of DNA which is formed new is depending upon the medium in which the bacteria is grown.
Oh, okay, got it.
ऐसे करने के बाद ये कंफर्म हुआ कि DNA replication is semi-conservative. This was
semi-conservative.
Wilson and Stahl, and it was proved that this is what happens.
Oh, wow. Okay, got it.
अच्छी तरह समझा DNA replication?
Yes, अच्छे से समझा. Yes, no doubt.
Seen and we have understood one of the autocatalytic functions of the DNA, where a DNA molecule gives rise to another DNA molecule.
Oh.
सब में जो भी steps में जो भी enzymes के नाम है सब याद रखो.
Of course, yes.
So we have seen two things for today. One is about the DNA packaging in prokaryotic as well as eukaryotic cell, and the other thing, the DNA replication, semi-conservative method.
Yes, correct.
Many things we have seen for today. Okay. Next,
yes.
Begin with a very important part of molecular biology that is called as the central dogma of life, that is protein synthesis.
Central dogma, yes.
You have learned some part of it in the 10th standard, but 12th not detail. We have to learn it separately.
Okay.
As well as eukaryotes.
Okay, got it.
So what I want you to do is, before I continue with this in the next session, 10th standard.
I will revise. Yes, of course.
So that we can build upon that on the 12th standard level.
Of course, yes.
Okay.
Yes, clear. Very clear.
Very important, and this is a very, very basic concept. Lots of possible questions out of all this.
Yes.
You cannot afford to lose touch with the topic in between.
Yes.
उसको अच्छे से, इवन आज का भी जो हुआ अच्छे से यहाँ तक अच्छे से रीड कर टेक्स्ट बुक में से.
Of course, yes.
Okay. So
yes.
No doubts.
No doubt.
Okay. So we will stop here for today.
Okay.